I think the text is going to be a little bit small because I was counting on a bigger screen, not on a TV. So if you can't read it, you can move closer. I am going to read probably most of the relevant parts, so maybe it's going to be okay. So let's talk about the gut microbiome. Why should you care about this? You should care because gut microbiome involves so many processes in your body. It generates thousands of enzymes for nutrient absorption, but also for physiological processes that involve things like blood coagulation and lots of stuff that is not directly related to nutrients. A large portion of neurotransmitters is created in the gut. It generally doesn't get into your brain because of the blood-brain barrier. But it does involve the brain indirectly because the gut is full of neurons, and the neurons are connected through nerves. I think it's mainly the vagus nerve that connects the gut to your brain and to the central nervous system, and the connection is bidirectional. So whatever happens in your gut can affect your brain and vice versa. The gut microbiome also modulates the immune system somewhat directly. I'm not sure about the specific mechanisms, but you've probably heard about it anyway. A properly functioning gut barrier is quite critical for health because it basically decides what kind of things from your gut get into your bloodstream and what kinds are blocked. But if you don't have any health issues, whether they are physiological or psychiatric, because it also involves like mood disorders and stuff like that, you probably don't need to care about it. Like generally, if you feel great, then your gut microbiome is probably okay. So I'm gonna have to give you a disclaimer because my education is in computer science and not in the microbiome, not in medicine or microbiology. I'm only interested in this topic because I'm a patient with chronic fatigue syndrome, which is probably caused by gut microbiome dysfunction. So that's the reason why I've been interested in this for some years now. And I don't have time to dig for sources because I only finished the presentation today. So if you're asking for a source, then the implicit answer is that it was revealed to me in a dream. I would like to thank this guy because this is a Canadian citizen researcher, basically, who discovered many of the things that I will be talking about today. He also suffers from chronic fatigue syndrome, and he's a mathematician, statistician, who also has Asperger's syndrome, which he says helps him focus on complex mathematical systems. And he wasn't the first person to associate chronic fatigue syndrome with a gut microbiome dysfunction, but he was, I think, the first and maybe only person who got the idea to collect thousands of samples of gut microbiome sequencing from healthy and sick people and do large-scale advanced statistical analysis on them to seek out some interesting patterns. Okay, so I see that this is really very small. Unfortunately, I can't make it bigger. So the question, how do you know that you have a problem with your gut microbiome? In fact, gut microbiome is called microbiome dysbiosis, which just means that the gut bacteria and viruses and other organisms cause stuff that is unwanted generally, that is detrimental to your health. It can be diagnosed by gut microbiome sequencing. However, sometimes people who objectively have a dysbiotic gut don't have any health issues. That is probably because they still have very well functioning gut permeability, which means that they have pathogenic bacteria in their gut. However, the pathogenic bacteria cannot get into their bloodstream, cannot get into the rest of their body, cannot activate the immune system too much, so they don't have any issues. We're not exactly sure why this happens, but it seems to happen, so it's a complicated thing. Digestive issues with gut microbiome dysfunction may or may not happen. You may not have any typical gut issues that you would expect, like diarrhea or constipation or stomach pain or things like that, even if you have gut dysbiosis. There's a funny thing called Bristol stool scale, which is an image with different illustrations of different types of excrements, where you can basically identify whether you have healthy shits or not. I'm not going to show it here, but anyway, it's not really a decisive thing. If you have a gut microbiome problem, you may have negative reactions to food, supplements, or medicines, but you may not, again. I think a good clue that you may have an issue with your gut is when you get health problems after events that disrupt your gut. So taking antibiotics, having bacterial infections or viral infections, for example, COVID is really, really bad for your microbiome in the long term. And if you react like that, then there is a good chance that you probably have an issue. The issues may be very different. It may be things like brain fog, problems with concentration, problems with memory. It may be physiological. It may be diarrhea or gut pain or stuff like that. But it may also be depression or anxiety. That's also often related to the gut microbiome. And generally, when you have a suspicion that you have a gut problem, then you have to explore all the traditional options. You have to check everything with standard doctors. And when you have, no other issues found, and it seems like it may be microbiome, then it's a good idea to explore it. But you always have to do the traditional medicine first. Okay, so the most important probably feature of the gut microbiome is that it is kind of stable and flexible. If you disrupt it just a little bit, so you get a sore throat and you get antibiotics and you take antibiotics for 10 days, then it will damage your microbiome, but in some time, it may be, I don't know, weeks to months, it will generally go back to normal because normally your gut microbiome is in a state of sort of equilibrium where different bacteria support each other or keep each other at bay, fight for food or kill off each other, etc. And it tends to get into sort of a local minimum as in the image and stay there. And if you disrupt it just a little bit, you will push it away and it will generally get back into the stable state where it was, or some different stable state, but a healthy state nevertheless. However, when you disrupt it a lot, for example, by taking antibiotics, getting COVID, and having a bad diet, you can get out of the local minimum, and you can basically push it away into a new local equilibrium, which can be a bad one. Now, this is an issue, of course, because when you get to a new equilibrium, it is very difficult to go back. So for a healthy person, usually you don't have to worry for like just taking antibiotics or whatever. But when you have a chronic stress and you have to take antibiotics for a long time or whatever, it can create problems that are very, very difficult and take a long time to treat. And in reality, I have drawn these images as like sort of simple two-dimensional illustration, but in reality it's probably more like this, where it's a very difficult 5D scale and you have no idea where you are and you have no idea where to push the microbiome back to make it better. So yeah, this is why a course of antibiotics normally doesn't hurt you too much, but this is also why when you do have a gut microbiome problem, you're not going to cure it by just taking a round of probiotics or eating fermented foods, or even a healthy diet doesn't really help too much usually when your issue is bad enough, because it's not enough to give the microbiome a push and push it to a different, healthier equilibrium. The question is also, what is a healthy diet for your gut? We are not really entirely sure, but it seems like the healthiest diet is just like what you would expect. Healthy whole foods with lots of diverse sources of fibers, so vegetables, fruits, pulses. Grains are not really a problem, meat is not a problem, but like avoid processed foods, avoid alcohol, avoid chronic stress. Chronic stress is really bad for your gut. It does alter your gut microbiome. And this usually doesn't work as a treatment, but it does work as a prevention. So start now if you don't want to have any issues. So let's talk about probiotics, because that's a very common popular topic and it's kind of complicated. They don't really help often, but they also can help, but we'll get to that. If a doctor just tells you to go buy a probiotic when you take antibiotics, that's kind of really stupid things to say. It's kind of like when your employer would tell you go buy a vehicle and they don't tell you whether the vehicle should be a tricycle, a trailer truck, or a helicopter, because there are many different types of probiotics. They each have different functions, sometimes very, very different functions, and some can even hurt you in some cases. Most of the probiotics you are going to see in a pharmacy are a mixture of what used to be lactobacillus. It's strike through because lactobacillus doesn't really exist anymore. The taxonomy of bacteria changes all the time. So what was called lactobacillus is now called lactoplantibacillus, lacticacebacillus, and some third one I don't remember. So it's stupid. But it's usually a mixture of those and bifidobacteria. They often don't really help with anything. And there are many more very different bacteria in commercial probiotics with very different functions. For example, Germany produces several types of E. coli probiotics. You may have heard of E. coli as a pathogen, but it's also a very important probiotic bacteria. It's one of the first types of bacteria that settles the gut microbiomes of infants. And Mutaflor is for some reason prescription only, even though it's freely available in Germany. Symbioflor is another product that can be purchased here through Amazon. Mutaflor was a probiotic that was used to cure diarrhea of Adolf Hitler. So yeah, it's famous. Japan uses a completely different type of bacteria, Clostridium butyricum, which again, Clostridium is known to be a pathogen, but it's also a probiotic bacteria. It also has been isolated nearly 100 years ago, both of these. I think in like 1933, so it's very old. It's also very popular, and it works completely differently. Probiotics can hurt, like I said. Just recently, I think like 2 months ago, an interesting study came out which found out that Lactobacillus gasseri helps Clostridium difficile infections, which is a relatively common and relatively problematic gut infection, whereas Lactobacillus acidophilus makes the infection worse. It helps the proliferation, increases the number of Clostridium difficile bacteria, and makes the symptoms worse as well, I think. And in reality, if you go to a pharmacy, you are much more likely to receive Lactobacillus acidophilus bacteria, which will make it worse. However, some of the probiotic mixtures contain both of these. So yeah, what is sold right now is kind of stupid. Another issue is the quality, because quite often when you buy commercial probiotics, the quality is going to be terrible. Half of them are going to be dead, sometimes all of them. And also some of the studies that tried buying commercial probiotic mixtures and sequencing them found out that quite often you don't even get the bacteria that are on the label, and instead you get some different bacteria because of contamination during manufacture. Sometimes even dead bacteria can induce changes in the gut, but it doesn't apply to all the probiotics, only to some. For example, when the function of the bacteria is that it directly modulates your immune system, it stimulates your white blood cells. That can happen even with dead bacteria because the blood cells react to some fragments of the cell wall, for example. So in that case, even dead bacteria work. Okay, so when can probiotics be beneficial? Probiotics can supply bacteria with manufactured enzymes. So enzymes are very important. And if you know what kind of enzymes you may be likely missing in your gut microbiome, which is theoretically possible these days, then you can get probiotics which will supply those. The issue with most probiotics is that they do not colonize your gut. So as long as you are consuming them, they work. And when you stop consuming them, they basically get out of your gut in like 1 or 2 weeks, and they stop working. So this is an issue. However, there are sometimes probiotics that do colonize. For example, those mentioned German E. coli probiotics, they do colonize, and they have been detected in your gut up to a year after you stop taking them. But generally, it doesn't happen. Many probiotics produce bacteriocins, which is basically a type of antibiotic. The bacteria use them to fight other bacteria, basically. They evolve to create antibiotics to kill off their competing bacteria in the gut. And this is very beneficial because you can take probiotics which will kill off pathogens in your gut. They can also have symbiotic relationships with other bacteria, so they can produce metabolites which will feed other bacteria and increase their numbers. Both of those things can happen. And these things induce long-term changes because even if the bacteria do not colonize your gut, so you will basically shit them out after a week, when you take them for 2 weeks and they produce antibiotics in your gut that kill off other pathogens, then usually other parts of the microbiome will fill their place, and it may produce long-term changes. The gut microbiome may stay like this even after you stop taking the probiotics, which is very useful. However, it has a problem. As with normal antibiotics, you can induce antibiotic resistance in those bacteria. So when you use probiotics that generate bacteriocins, which kill off other bacteria, and you take them for a month or two, after, say, 4 or 6 weeks, they may suddenly stop working, and you may start feeling much worse. And that is because you basically supplied small amounts of antibiotics to your pathogenic bacteria, and you killed off most of the pathogens, but not the ones that, the small amounts that evolved resistance, and then they started to proliferate. This is the same reason why you shouldn't be taking antibiotics, normal antibiotics in pill form, in other ways than what the doctor recommended. So that can happen. Usually to avoid this, it is recommended to swap probiotics in like 2 or 3 weeks. Do not take them for any longer than that. Which again, the doctors generally don't tell you because they don't know, but yeah, that happens. Okay, so where to buy probiotics, and the ones that could possibly work, ones that are more suitable than what is in the pharmacy. You can generally buy probiotic powders online. It can be kind of expensive. Usually the price is like 30 euros to 90 euros per 100 grams pouch of like pure powder. However, if you count the price per colony forming unit, which is a standard unit of like the probiotic amount, then it's usually cheaper. Also, you can buy the powders that have been manufactured like 3 weeks ago, so you know that they're fresh. They haven't been lying on a shelf for like 2 years somewhere. Dosage matters a lot. Quite often, if you look at studies that talk about using probiotics, they recommend taking like 3 times or 5 times higher dosages than what is recommended on the pills that you buy in a pharmacy. There is a problem with patenting probiotics, and that is because you can patent probiotics, you can protect them, and so it is also relatively easy for a big biomedical company to create genetically novel bacteria. So what they do is that they develop novel bacteria that are only very slightly genetically different than existing bacteria, so that they can apply for a patent. And then they run studies on these specific strains, so that if it turns out that the probiotics work very well, they already own the patent and they can start manufacturing them and selling them for very high prices. The problem is that when you open like some popular science website and you read about some great novel bacteria that help with long COVID or whatever, and you're excited to buy them, generally they are not on market anymore, or they are not on market yet, because it takes some time to go through all the clinical trials to sell them to the public. And if they are on market, they tend to be incredibly expensive. And another issue is that quite often a similar strain that is cheaper or not protected by patents anymore would work exactly the same, because the genetics that are important, that are relevant to the function that is being studied, is exactly the same. However, we can never really be sure that that is the case. So if you see some specific strain of Lactobacillus acidophilus, you can never be sure whether the one that you buy here in a pharmacy is going to work the same, unfortunately. So yeah, patenting is kind of, it sucks. Yeah, a lot of published research is kind of useless because of that. Also, another issue with probiotics is that they induce different effects based on what your microbiome is at the time when you start taking them. 2 different people with 2 different microbiomes take the same probiotic, and each of them is going to have a different result, different effect. That is the unfortunate fact, which also makes research complicated. Choosing the right one is I think the best way to choose the right probiotic is just to do microbiome sequencing, which we will talk about later. But the real answer is that there is no really great, sure answer. It's always kind of hit and miss. You just have to try it. And the rule of thumb is that if you buy a probiotic, you take it for 3 days at the recommended dosages that are recommended not on the package but in the studies, and you feel no effect, then it's probably useless. Whether it's just not suitable for you, or whether it's dead or whatever, but it should do something. Like it's not normal that you buy a medicine in a pharmacy and you take like 4 times the recommended dose and it does absolutely nothing. That's a good message that it probably doesn't do anything for you or in general. Okay, some practical tips. It's probably not a good idea to take probiotics after you take antibiotics. Surprisingly, it can help with digestive issues if you have digestive issues from taking the antibiotics. But it seems like from several studies that it slows the recovery of your microbiome. And it's better to just leave it be. Eating fermented foods is probably okay because fermented foods provide relatively diverse amounts of bacteria. But probiotics are probably bad for you after you take antibiotics, it seems like. You should know about Saccharomyces boulardii yeast, which is probably the most universal pill against pathogens that cause like stomach ache and diarrhea and stuff like that. I recommend you buy it and take it with you if you go abroad, because I think it works better, more universally than lactobacillus bacteria. And again, dosage matters because a very popular form that is sold in Czechia is called Enterol, and it has like 1/5 of the dosage that is usual in other pills. So yeah, check the dosage. I think Dr. Max Pharmacy sells it as Enteromax Travel, and it's really useful. I really recommend you have it. Every time a person around me has a problem with stomach pain or diarrhea or whatever, this thing seems to work. Lactobacillus and bifidum mixtures usually don't work that great for diarrhea or stomach pain. They are quite selective on the types of pathogens. And if you have some long-term gut issues, including stuff like irritable bowel syndrome and stuff like that, then the German E. coli probiotics like Symbioflor 2, which can be ordered through Amazon or through German online pharmacies, it's a pretty good bet that it might work. I would recommend you try it even without sequencing or anything. Yeah. And taking one strain of bacteria is usually better than a mixture, if only because it allows you to cycle through the different probiotics so that you don't develop a resistance, basically. You might have heard about fecal microbiome transplants, or FMTs, which is a very tasty process where other person's shit literally gets implanted into your gut. It happens either through eating shit capsules, which is not that common anymore, or I think usually in Czech hospitals it is done through a plastic tube that is shoved to your nose and through your stomach into your upper intestine. And then the healthy person's shit is taken out of the freezer, it is unfrozen, it is mixed in a shit blender with, like they literally have a dedicated blender for blending shit, mixed with a physiological liquid. It is taken with a syringe, and the syringe is inserted into the tube, and through the tube it gets into your upper intestine, small intestine, and sometimes it's also applied using an enema, which I think is the most common way that is for some reason not used in hospitals in Czechia. I don't know why, but yeah, these kinds of very tasty things. It is very useful for treating Clostridium difficile infections, which are difficult to treat because they are basically caused by antibiotics. When you get broad-spectrum antibiotics for something, they kill off most of your gut microflora, and they create basically they create a power vacuum in your microbiome that gets taken over by pathogens. And one of those pathogens is Clostridium difficile. It's very common. Most of us have some amount in our guts, but when you kill off most of your microbiome, then it proliferates and causes quite nasty infections. And because of this, you can, of course, treat it using antibiotics. However, when you use antibiotics, you again kill off most of your other healthy microbiome. and usually you don't kill off all of the Clostridium difficile bacteria, so basically the same thing can happen again. It again overgrows and returns and returns. So we have discovered many years ago that fecal microbiome transplants are a very successful treatment for this, because they refill your microbiome with healthy bacteria and allow it to create and basically give it time to create a new healthy equilibrium. Fecal microbiome transplant theoretically can be used for other more complicated illnesses like chronic fatigue syndrome, long COVID. I'm not sure about Crohn's, but certainly irritable bowel syndrome and stuff like that. But it has two issues. The first is that it is surprisingly unpredictable. There has been a study which did microbiome sequencing of the healthy donor whose shit is used to implant into somebody. Then they sequenced the microbiome of the recipient, of the patient, before the implant. And then they sequenced the resulting microbiome of the patient after the fecal microbiome transplant. And what they found out was that basically the resulting transplant is definitely not an average of those two microbiomes, the donor and the recipient, and instead it's completely different. And there were many bacteria detected that were not detected in the previous pre-transplant microbiome of the recipient and the donor. So something very weird happens, and we're really not sure like what's going to be the result. No?
How do I become the donor? Do I have to register somewhere? Okay, I would like to donate my shit.
Okay, if you're super healthy and you don't take antibiotics and stuff like that, yeah, that's a problem, yeah. It's like, I think the criteria are quite strict, but I'm not sure, because I am the one that's sick, so I didn't really look into the option of donating. But
if you ever want my shit, I'm here for you.
Yeah, okay, I can ask. My friend is a doctor who worked in one of the hospitals in Czechia where they started doing this, so I can certainly find out. Yeah, sure.
I'm selling my shit for cheap.
Actually, there is kind of a black market for healthy shit because people do this DIY when they are very desperate, which I think it's a bad idea because it is dangerous. You can make yourself worse. You can give yourself illnesses and stuff. It is a dangerous thing to do, but desperate people do it, and sometimes the result is bad. Yeah. Yeah, South Park has an episode about fecal microbiome transplant, and it is very accurate. So yeah, it is actually good to learn about this stuff. Okay, and another issue is that when you are treating just a Clostridium difficile infection, just one microbiome transplant is enough. It's okay, but with more difficult illnesses, you need to like really shift, really push the microbiome to change. And one fecal microbiome transplant is not enough. Usually, studies that are successful in treating even things like symptoms of autism. Autism seems to be linked to the gut microbiome. They usually use like 10 or 20 or 30 microbiome transplants every day for a month. Sometimes they also use high doses of antibiotics before that to empty out the recipient's gut microbiome and stuff like that. Like it's quite difficult to do, and we're not exactly sure like what is the best method yet. And also choosing a healthy person is quite difficult as well, because unhealthy FMT can make things worse. In Czechia, I think it's officially only done for the Clostridium difficile infections and similar things, although there is some doctor's discretion. I've been told that they can use it for other things, but not for things like chronic fatigue syndrome, definitely not. There is a private clinic in Slovakia, in Topolčany, surprisingly, that does this, but it costs 4,000 or 5,000 euros, so it's quite expensive, and like the results are not guaranteed. Okay, so let's talk about microbiome sequencing. The first issue, there are several issues with this. The first issue is like, where do we take the shit, like specifically in a person that we sequence? Because the microbiome in your small intestine is different to in your large intestine. it's different in your colon, and it's again different in the feces that are outside your body, and they are reacting with oxygen and yeah, stuff like that. So ideally, you would like take the microbiome sample straight out of your body, and that's obviously not possible. So we only sequence the fecal microbiome. But this has another issue, which is that feces have bacterial hotspots. So there are like spots in your shit where there's like 20 times the concentration of certain bacteria. And it happens with like important bacteria like Bifidobacteria or Akkermansia mucinifila, which is a type of bacteria that is linked to good health and stuff like that. So again, yes?
So can you blend it?
Yes, a sheet blender would again be the best solution. However, that is impractical. Most of us do not have a sheet blender. So normally what happens is that the lab that I use sends you a sheet shelf called feces catcher, which is a strip of paper with glue that you put into your toilet bowl, and it basically acts as a shelf. And then you take a sterile swab, and you swab the shit in like different spots and different depths, etc., to create an average. And then you flush all of it because it's flushable. Then the sterile swab with the sample is put into a small vial, which contains a preserving buffer, which kills off the bacteria, however preserves their DNA, which is practical because it's not a biohazard, so it can be shipped abroad, and also it doesn't have to be refrigerated. It lasts for about a month or something. Another issue is when you collect the sample, because your microbiome changes based on the time of day, based on what you ate last few days, based on supplements or probiotics and whatever. So generally it's advised to cease taking antibiotics or probiotics and other supplements 2 weeks before that, and collect the sample in your first bowel movement of the day to create some consistency. Okay, then how do we sequence it? The first option is to use PCR testing, but the problem is that it gets very expensive very fast because you have to do each PCR test for each bacteria that you test. So this is good for certain pathogens because it is quite exact, and when you're testing for just a few bacteria, it's not very expensive. But generally, for our purposes, it is impractical. So normally, we do next-gen sequencing when we try to identify hundreds or thousands of bacteria. And there are two main methods. One of them is shotgun metagenomics, which is the best method. It identifies bacteria, viruses, yeasts, molds, everything. And it can identify generally like single thousands of bacteria in a sample. However, it is still kind of expensive. Like one testing costs like 8,000 to 15,000 crowns around here. So what is most often used is 16S RNA testing, which tests a specific segment of RNA that is only contained in bacteria, which means that this test only finds out bacteria. It can identify like 500 or 600 bacteria. It is not exact. It is kind of a guessing game. And it doesn't give you specific information about individual strains. So it gives you kind of a big picture view. But it turns out that this is good enough. And it is indeed what most scientific studies use to this day, I believe, simply because it's cheap. It can be bought for about 80 euros, I think. So there are several levels of like imprecision, of uncertainty. And the question is if it can even be like precise enough, if it can give you useful information. And statistical analysis says that it can. It can find patterns which can be seen in like published literature, which reproduces its findings about like different microbial patterns and different illnesses. And also statistical analysis on large collections of microbiomes from different people can also identify patterns linked to like sometimes very specific symptoms like trouble falling asleep or waking up groggy or having issues recalling names, stuff like that. So this shows that it's to some degree works even through some of the issues. Okay. Most of the labs I would like to mention, because like just to warn you in case you ever decide to do this, is the issue with the labs is that usually there is not a high cost margin on the sequencing itself. So their business model is that they try to upsell you. they give you microbiome sequencing, and then they try to sell you probiotic mixtures, prebiotic mixtures, expensive analysis, etc. So don't ever do that, because the science is usually very outdated and it's useless. Microbiome research is incredibly active, and that's the reason why it's useless, because it is incredibly expensive to have competent people which kind of read all the new studies and use them and kind of create actionable information, actionable outcomes based on that. I think it's still the most active area in biomedicinal research, and the number of papers is like, I don't know, like 50 new papers a day or something completely absurd like that. Like nobody can read it. And it evolves very fast, so anything older than like 5 or 10 years is completely useless. Okay, so let's talk about why I was thanking the old Canadian guy in the beginning and the project that he made. So what if we collected a few hundred to a few thousand results of microbiome sequencing, created a database from different people, and then told the people to label like their official diagnosis, like any illnesses they have, and also go through like big questionnaires with a few hundred questions about their specific symptoms, about like whatever physiological or mental symptoms, or very specific types of fatigue, very specific problems with sleep, or yeah, like whatever has been used in like different university questionnaires in research. And then we could run advanced statistical analysis based on that data. Turns out that even through the imprecision in sequencing, this gives us very interesting results. And even though the analysis was made on just very simple criterion, which is taking bacteria in each sample that are in a very high amounts or very low amounts. So basically, if you have more lactobacillus than 90% of other people, then that is deemed too high. And this criterion, like very simple, like percentiles or box plot whiskers, standard deviation, etc., were used for these analyses, and it still gave us very useful patterns for specific symptoms. There was an interesting thing. Sometimes this analysis would give us two very different microbial patterns for the same illnesses, which shows us that something is weird. Either the analysis is incorrect or it gets more complex than it seems. For example, with chronic fatigue syndrome, there are people who have almost no lactobacillus detected, which is me, or there are people which has like way too much lactobacillus, more than 90% of other people. And they both have like the same symptoms. So there is one very interesting Japanese project called Kyoto Encyclopedia of Genes and Genomes. It was done by Kyoto University, and they basically tried to create a huge database of genomes of all the available cells, including bacteria, and publish it as open data. And thanks to that, we can get a very rough idea of, for example, metabolites that a bacteria produces or that it consumes. It is not like super precise, but it can give us some sort of like big picture view. The sum of what the whole microbiome consumes and produces, it is called the metabolome. And when we look at the different microbiomes of people with the same illness that I mentioned in the previous slide, we can find out that even though their microbiomes seem to be very different, their metabolomes are very similar. So two very different microbial patterns can cause the same issues because they produce the same enzymes, the same endotoxins, the same whatever chemical compounds they create. There is not really enough data to say this with certainty because of all the uncertainties in the sequencing, etc. And because we don't have, for example, specific data about the amounts that of certain compounds that the bacteria produces. But it does seem like enzyme production is maybe the most important thing of the gut microbiome and not the bacteria themselves. Okay, so how do we use this? We can collect another data. We can look through all the published papers on how many different like food items, prescription drugs, probiotics, prebiotics, etc. reduce or increase the numbers of specific bacteria in your gut. Some of the studies are very good, some are not that good, but basically we can assume that if we put all of them, which is thousands, into a huge database, then on average we will get like some okay data. So if we do this, then the database allows us to ask the following questions. Which bacteria does, say, inulin, which is a prebiotic, increase or decrease? And which items should I start or stop taking if I want to increase like bifidobacteria or Akkermansia or other bacteria? If we put all of this together, then we basically have a method to gradually change the gut microbiome. Because we can now sequence the gut microbiome, get a list of all the bacteria that were detected and their percentages, their ratios. We can identify bacteria that are likely too high or too low based on several different methods. We can make a consensus list, basically. So if a bacteria is labeled as too high by several different statistical methods or by association to certain illnesses or conditions, then it's probably problematic. And if it's only labeled as problematic by one statistical method, then we can probably throw it away. So we make a consensus list and give high rating to bacteria that seems to be bad by more than one method. And then we can get a list of drugs, supplements, probiotics, prebiotics, etc., which should affect the problematic bacteria in ways that, that are practical, basically, that can fix the issue. Okay. The list can look something like this, and you can probably not read it at all from the distance, but anyway, it's a list that contains things like sugar beet, it contains some vegetables, it contains some like 5 different probiotics. It doesn't contain any antibiotics because I excluded them. It contains yogurt and yeah, some vegetables and stuff like that. It is sorted by priority, and the priority is based on how many bacteria is affected. Second, just a second. How many bacteria is affected in a positive way that we want, and also by how many studies is the effect supported. On the right, you can see a column that is in a red box. That is a link, which if you click, which we can do here because it's an image, but anyway, it's a screenshot of the analytical system. It will list all the published literature based on which the recommendation is made. So basically, you can check if the quality of the studies is good enough. Okay, question, time. We are getting to the end, yeah. What was your question?
I wanted to reference AlphaFold from like recent discovery of finding like 200 million how to fold a proteins. Like, are there any research into what you were doing with the Kyoto or the
Yeah, okay, I'm going to just repeat the question here. Don't need the. Okay, so the question was whether AlphaFold gave us some interesting results that would be applied to this. I think not yet. We will see. I don't think the Kyoto University uses this. I think it's just standard like shotgun metagenomics and analysis. Yeah. Okay. It will also, the system will also give you things that you should avoid. And the interesting thing is here that there are 2 things: iron supplements and slippery elm, which is a herb, which are generally recommended to treat chronic fatigue syndrome. So there's a good chance that I would just try to take them just in case it could help, or any other person. However, in this case, it would probably hurt and not help.
Sorry, did you say a herb?
Yes.
A plant, okay.
A plant, yes, yes. So the whole cycle is that you test your shit, you get recommendations for things that you should take or avoid. You take like the top 20 things or something like that. You will make like 2 to 4, you will divide them into 2 to 4 groups at random basically. And then each week you focus on one of the groups. So like five recommended items. And you do this so that you avoid the bacterial resistance that we talked about. So you say you take 2 different probiotics, 2 different prebiotics, and some supplement for a week, and then you start taking something else, and you do this for like 2 months, and then you retest, see whether your microbiome changed in a way that was expected, And if it did, then you test your shit again and make a new plan and do it for another 2 months. And theoretically, in like 6 months to a year, you should have a considerably healthier microbiome without the risks of fecal microbiome transplants or drastic diets and stuff like that. And as far as I know, this is basically the state of the art in relation to analyzing and treating the gut microbiome. It does work for some people, it doesn't work for others. We're not sure why. It may be that the method abstracts away so much complexity that some issues are just not visible. It may be that people don't do it properly enough, we don't know. But anyway, the author, Ken Lassesen, who made this system, has had success with this because he has had, I think, 3 different relapses of chronic fatigue syndrome, which unfortunately is a thing that happens. And each time he was able to get into remission faster. Okay. And there is one very interesting thing that I have to mention. That basically is some evidence, again, that it works. The database allows us to basically ask the question, I have a certain illness, I would like to, that probably affects my microbiome, I would like to treat it, what should I take? And it turns out that in many cases, this answer gives you the same medicine and supplements that is recommended for treating the illness by standard medicine that is completely unrelated to the gut microbiome. So it turns out that some of the medicine that the doctors will prescribe you works on your microbiome in an advantageous way, basically coincidentally. We are not sure why, but it may explain some of the unexplained effects of certain medications like antidepressants, etc. Okay, so even though this was already way too long, there are still many things that did not fit because it's a huge, complicated topic. So again, if you have some questions, we can talk about it later to not delay this anymore. There are some random stuff like, for example, you can look at all the compounds, at the metabolome, at all the compounds that are produced in your gut. And for example, you can find out that your gut creates ethanol, small amounts of ethanol, which can give you non-alcoholic fatty liver disease. So if you suffer from fatty liver disease, even though you don't drink any alcohol, this can be the cause. And there are many different things that you can analyze using this system. However, you need to have the medical knowledge to do this. It is very, very complicated. And yeah, okay, I'm not going to mention the rest because we're out of time. But yeah, that's it. Yeah. Okay, question?
So I'm vegan, so this I would like to reference an experiment from Justin Otkin from the Thought Emporium channel, where he, in summary, he has a lactose intolerance, and he injected himself with adeno-associated virus to make his gut produce beta-galactosidase enzyme, so that he's able to then eat lactose things without ending up in agony. Are there any similar, when I say it, bioengineering mods that you would recommend to a vegan?
I only recall one study which is great. I love it because it's really funny and disgusting, and it is actually on the same topic. The United States of America has huge reserves of powdered milk because they subsidize milk farmers. And they use this powdered milk to food help in third world countries, in Africa and places like that. And the problem is that people in Africa usually don't have the gene that allows them to digest lactose in adulthood. And so Americans found out that if you feed people who are lactose intolerant huge amounts of lactose, then they suffer a lot for 2 weeks, and after just 2 weeks, basically their gut microbiome grows enough bifidobacteria to digest lactose that it's no longer a problem. And I have seen a video from some American biohacker who tried this thing exactly. She had lactose intolerance and decided to shovel huge amounts of powdered milk into herself for 2 weeks. And she said that the stomach ache was terrible and the farts were absolutely horrible. Her roommates totally hated her for 2 weeks. But really, after approximately 2 weeks, she was able to start digesting lactose again, and she's fine. So that's it. But I am not like specialized in this. I mostly research things that were relevant to me because like the field is already huge. So this is the only study that I remember.
I meant like, are there any bioengineering modifications that you would recommend, like generally? I just use this like as a reference to what I saw is being done. To like, are there any other things that would be useful?
This is not really like the field that I was researching, so I don't know. I don't have the answer.
I feel healthy. I don't have any symptoms, but I eat a lot of probiotics because everyone say it's healthy. So could this really damage my health?
Probably not, but it probably doesn't help either.
Okay.
Like I would suggest you eat like a lot of different vegetables and fruits and pulses like beans and stuff like that. And that's probably a better idea than buying probiotics. Okay. Again, I'm here for the night, so if any of you have any other questions later, then feel free to ask me. And thanks for your attention.